So today we're going to demonstrate a few techniques which can be used to demonstrate fungi microscopically. The first one is called a sticky tape prep, and this is the one that's used most commonly in the lab. Just add a few drops of stain to your slides. Use common everyday clear sticky tape. Just take a small bit of sticky tape and an orange stick, and you make what's called a flag. Then, using the flag, put the sticky side down onto the fungi that you're interested in. Just press very gently, and you can see that there's some fungal spores and hyphae have attached to that sticky tape. Then, just using a drop of 95% alcohol, turn the flag over so that the sticky side is facing up, and just add a drop of alcohol so that it detaches, and then just lay it in the stain. You then add a cover slip and it's ready to look at. Now, this particular fungi that I just did is quite granular, so you need to be quite gentle when you make a sticky tape with this fungus. But if you have a fungus that's more fluffy, like this one here, then you need to, when you make your sticky tape, press more firmly so that you can ensure that you get some spores on the sticky tape. So again, make a flag. Touch it to your fungus. Just bend your stick back over so that you can press it quite firmly down onto the, and probably I've got too much now, but that's what you do if you can't get many spores attaching to your sticky tape. Add your drop of alcohol and just lay it down in the stain. So the advantage of the sticky tape is that it sticks the fungal hyphae to the sticky tape and just locks them in place so that they look exactly like they would look when they're growing. And this stain that we use is called lactophenol cotton blue, and it's got various components that make it safe for us to handle the fungus. The cotton blue is a basic dye which gives the fungi it gives the preparation a bit of colour, so it enhances the contrast of the structures. The lactic acid is a clearing agent and helps preserve the fungal structures. Phenol kills the fungus and makes it safe to handle, and glycerol is slightly viscous and prevents drying of the prepared slide. Now, the advantage of this sticky tape preparation is that you can also use it directly on food. So if you have a tomato or a bit of bread that comes out and it's got these like furry bits on it or black heads that are the spores, you can use your sticky tape directly on that piece of food. So that saves you a bit of time. You don't have to actually grow it. The next technique that we're going to use today is called a tease mount, and we generally use this for zygomycetes. These are the very rapidly growing fungi that often lift the plate on the Petri dish after 3 to 5 days, and you can see that there's some spores in the lid of that plate already. So with this technique, we usually add a drop of alcohol first, and the alcohol acts as a wetting agent and drives out any air bubbles that are trapped between the hyphae. Just pick a bit of hyphae, and you can see it all sticks together in a big clump. So put that into your alcohol, and just with another needle, just tease it out so that you can see the structures. So this is why it's called a tease mount. With the zygomycetes, because the hyphae is very robust, when you do a sticky tape, you often end up with only hyphae and no rhizoids or fruiting heads. So So once you've teased it out, you can then add a drop of stain and your cover slip. And then that's ready to look at under the microscope. The next technique we're going to use is called a squash mount, and we usually use this technique when we have very glabrous fungi. Now, glabrous is a word that means skin-like or leather-like or without hair, and when we use it to describe fungi, we usually mean fungi that are quite, sometimes they can be skin or leather-like, but they're like this one, quite smooth and glossy. So just take a small amount of the fungus, mix it up in your stain, add a cover slip, and then just press down. And the reason that we do it for this particular fungus is because it's so smooth and glossy, it's so wet looking that it doesn't actually stick to the sticky tape. So that's the way we do that. There is another way that we can do a squash prep. So this is the same black yeast as we looked at previously, but it's a bit older, and you can see it's even lost its yeast amount, yeast look about it. It's quite glabrous. So what we do with these particular fungi is just take a very fine slice through the fungus. And you might need a little spatula to get it out. And just lay that down sideways in your stain. Then you add your cover slip and just gently squash it. And if it slips out from under the cover slip, you can just put it back on top. And you might need a bit more stain. Now, when you look at this under the microscope, it can be very thick where the agar is. So you start where the agar is and just move forward till you get to the edge of the fungus, or where it would have been the top of the plate, and then just focus up and down. And coming out from that surface, you should be able to see fruiting bodies or any characteristic fungal hyphae that's available. The final technique that we're going to look at today is called a slide culture. In this technique, we actually grow the fungus onto a cover slip and then we harvest the cover slip so that we can see the fungus exactly the way it grows. So take 2 agar plates, and one of them is just for your agar blocks. So just make a few cuts. Remove those agar blocks and put them on the other plate. And you can do 3 or 4 on the one plate. Then, using a sterile needle, inoculate the sides of those agar blocks with the fungus that you're interested in. And I don't know if you can see that, but there's some little green spores. adding to the side of those agar blocks. We then take a sterile cover slip and add it to the top of each block. And of course, when we do this technique, we use complete aseptic technique. And we usually do it in the biohazard cabinet so we don't contaminate our sample with environmental fungi. So then we put the lid on and incubate it upside down like that. And if it's a rapidly growing fungus, usually you'll get something growing on the cover slip in 3 to 5 days. If it takes longer, you might have to keep it for up to 3 weeks. I've got one here that we've grown previously, and you can see that the fungus has spread all over the plate and it's grown up the side of the agar blocks, but it's also grown onto the cover slip. So these cover slips are now ready to harvest. Now, for this particular technique, we use lactophenol cotton blue again, but it's a slightly different formulation. It has some PVA added to it, and the advantage of this is that we can make a permanent preparation. So you add your drop of stain and just harvest your cover slip. And I don't know if you can see that there's fungi growing on that cover slip. So this preparation can be looked at straight away, the same as the others that we've made. But if you just leave it on the bench for 7 to 10 days, the PVA cures and you get a permanent preparation that you can look at another date. And we've got some here that we've made previously, and we've made these for the class that you're having today. And this one you can see that there's where the agar block was, and there's fungi just growing around the edge. And this is another one where the fungi is all grown in from the edge onto the cover slip. So hopefully that will be